All right, good afternoon, everyone. No, just need that one. Okay, cool. Good afternoon, everyone. I hope that you guys are having a good start to your Thursday. It's day 3. I've done a couple of things. One, I've got our presentation for tomorrow as well up in addition to today's. Tomorrow you're going to have a quiz. That quiz is going to be, I think what I'm going to do, just because there's potentially some like hiccups that might occur, is that will be posted after our class is done, and that will be administered on Canvas, but I'm not going to introduce the variable of Proctorio. Our exam on Monday, we are going to be using Proctorio. Any directions on that I'm going to be adding, which reminds me, I forgot to add the top hat. And that means I have to extend the deadline. Top hat. I apologize for that. Code. Okay, so I have to add the top hat code. Full disclosure, the reason why top hat's kind of, I'll venture to say clunkier for myself than other instructors is that I took a class here at Texas State, and not whenever I was a student, but instead in my current role as an instructor. And so I had to have a separate account so that I can be top hat as a student, or be on top hat as a student and be on top hat as an instructor. My one account cannot do both things, so I have to have this other separate account, which I know sounds very fancy and very advanced, but it's really something that seemingly is a simple problem. At any rate, I apologize for that. I'm gonna give you all the top hat code. It doesn't, so when y'all go and click the top hat thing on the left-hand side, it doesn't say like, this is the class that you want to join. Is that correct?
I did it today and it finally like worked, and then I logged in with the Texas State. Like there's a button that says login with SSO, and it's all Texas State, and then once I log in with that, it just says there's nothing on there right now, but it works now.
So it doesn't show Chem 4375, it doesn't show,
it doesn't show any questions. It's just
okay. There should have been questions available yesterday at 4 p.m. that are due, or yeah, that would be due.
There is 3 questions that show up there that's due tomorrow.
Okay. Okay, well then, I guess what I'll do is I'll check on, okay, well, I'll go back and look at and give kind of a little bit of a grace period just as we're working through any sort of clunkiness or funkiness of my own. So in the event that you missed top hat because you forgot to do it, well then bank error in your favor. And so I'll be a little bit flexible with those deadlines and everything like that, just because I want everyone to be able to do them. With that said, I added questions corresponding to our Tuesday lecture. And those I've set at this point, so I'm kind of reverting back to what I said previously, where, oh, I'm not going to have any questions. I've added questions that correspond to Tuesday's content. Those will be available later today, and those will close on Saturday. So I'm going to give a little bit of an extension on those. Now, with that in mind, the top hat questions, if you look at them and you're like, man, these are easy. Yeah, they are. Like I said yesterday, you should not expect those to be, you should expect most of the top hat questions to basically be that easiest third sorts of questions. Now, with that in mind, I want you to use those as your means to.
I ain't eat nothing yet.
Can you mute yourself? Because I seemingly cannot mute you unless you're trying to talk, Mia.
Oh my fucking god.
Okay, cool. So I've added also to the discussion board on our Canvas page, so I would recommend just kind of dropping in with a question of your own. So try to synthesize some of your own exam questions and kind of, we can peer review those and yeah, I'll add some to try to get, I want that to be basically our study guide. As like, this would be a good sort of question. And I'll try to drop in and say like, yeah, this would be I consider this to be a challenging question. I consider this to be a mid-tier question. I consider this to be an easy question. So that's what I'd like to do, which I added to the Canvas page or our discussions earlier today. Now, with that in mind, am I recording? Yes, good, I am. I am. Okay, it says, yeah, okay, cool. So what we're going to do today is we're going to finish up our discussion of sugars, and we're going to use that as our means to segue into a discussion of nucleic acids and nucleotides. So when we look at a monomer or a monosaccharide shown here on the left, the example that's provided is ribose. Ribose, because it has a free anomeric carbon, is classified as a reducing sugar. So any sugar with at least one free anomeric carbon is going to be considered a reducing sugar. Monosaccharides that I can think of at the moment, all monosaccharides are reducing sugars. Now, when we think about a disaccharide, we've got a total of 2 disaccharides that are indicated here. We've got sucrose and lactose. Lactose is an example of a reducing sugar because lactose has a free anomeric carbon. Sucrose, on the other hand, is a non-reducing sugar because it does not have a free anomeric carbon. Instead, it has 2 bound anomeric carbons. So when we look at something like a non-reducing sugar, you should identify within each one of those monomeric units, each one of those monosaccharides, where's my anomeric carbon? And then say, all right, what is that anomeric carbon bound to? Is that anomeric carbon bound to another anomeric carbon, or is it not? I think that it's helpful to, when you look at a sugar, a disaccharide, try to break it in 2. Try to look at this right here, this individual subunit. Now, as you can see there, we have a oxygen, carbon, oxygen. Now that oxygen, the second one that I'm pointing to, when we're splitting this into 2 individual sugars, you should imagine that this is a hydrogen right here. Now that right there shows me that I have a hemiketal or hemiacetal. So your hallmark for a hemiacetal or a hemiketal will be O within my ring, going to a carbon, going to a hydroxide group. So the difference is that, so it's oxygen, carbon, oxygen. One of those oxygens is a hydroxyl group. The other oxygen is bound to 2 independent carbons or 2 separate carbons. Does that make sense? So whenever you're trying to figure out, oh, where is my anomeric carbon in a disaccharide, I think that's a very helpful thing to do. Now, in addition to that, well, we've got this other sugar right here. Obviously, we're breaking this bond kind of as like a visual cue. So we want to have OH. H. There we go. Got it. Kind of. Oxygen, carbon, hydroxyl. There would be my hemiacetal or my hemiketal. Does that make sense? So that's one of the things that you should be looking for in order to identify that anomeric carbon within a disaccharide or in a cyclic sugar. Okay, so identifying and classifying a sugar as a reducing or a non-reducing sugar, that's basically what your cue is. Now, when we look at a disaccharide that is a reducing sugar, we have our OCO. There's that hemiacetal or hemiketal that reacted with that right there. This carbon number 4 that has my hydroxide group going down. So now what we've got is that group right there. that constitutes a linkage between my two sugars. So these linkages are hallmarks of polysaccharides. We just looked at a disaccharide or a pair of disaccharides and classified those disaccharides as being either reducing or non-reducing sugars. All of these polysaccharides can be classified in that way, as well as adding a couple of components to it. Now, your most common polysaccharides are cellulose, glycogen, amylose, amylopectin, and chitin. It's important for you to know the differences between these and the ways to distinguish them from one another. Cellulose, what's cellulose a major component of? Plants. Now, cellulose is a repeating monomer or repeating polysaccharide of glucose. So it's glucose after glucose after glucose after glucose. It's D-glucose that just repeats over and over and over again. Now here what we have is a depiction of a fairly complicated oligosaccharide, where we have multiple subunits that make up a single component or a single molecule. Now, our polysaccharides, glucose or cellulose, glycogen, amylose, amylopectin, those four are primarily based on glucose. So cellulose is great because, oh man, it's just a bunch of glucose, but we can't eat it and actually extract any energy from it. And the reason for that is because of the linkages that connect each one of those individual subunits. So here is a cellulose molecule. One of the things that I want you to notice about it is this thing right here, this bracketed unit just repeats over and over and over again. Now, this N is our indicator of that. What that means is that you don't see any branch points coming off of carbon number 6. You don't see anything coming off of carbon number 2 or carbon number 3. Everything is between our anomeric carbon, carbon number 1, and carbon number 4. Does everyone follow that? So cellulose does not have any branch points. With that in mind, though, I want to go ahead and clear all of this and make a note of, well, what are each one of these? Each one of them is glucose. We got carbon 1, 2, 3, 4, 5, 6. We looked at a pattern yesterday for our cyclic glucose and we said down, up, down. Does this follow that down, up, down pattern? Yes, it does. When we look at carbon 2, our hydroxide is down. 3, our hydroxide is up. 4, our hydroxide is down. Now, it's not a hydroxide here, however, because what we've got is we have a linkage between carbon number 4 and carbon number 1. Now, carbon number 1, when we talk about glucose in its cyclic form, what is carbon number 1 also known as? It's an anomeric carbon. It's a newly formed chiral center. When we talk about anomers of two sugars, what are the two possible anomers of any sugar? Alpha and beta. Those are our only two designations. Now, with that in mind, if we have this anomeric carbon right here, and right here, and basically our hydroxide group is pointing upward, and carbon number 6 is also pointing upward from our ring, what does that mean about this particular sugar? Which anomer is that? Is that the alpha or the beta? The beta. The beta designation. Now, one thing that I want you to recognize that distinguishes biochemist from chemist is this depiction right here, this bond. Now, if you showed this to an organic chemist, and if you think about the rules that you learned in organic chemistry, whenever you see a point, what does that usually indicate? A point like this right here. There's a carbon there. Now, this is where biochemists have taken some creative liberties, and there's not a carbon there. This is instead a carbon-oxygen bond. Sometimes you'll see it drawn like this, which I gotta say, I think the people who are responsible for Metallica's logo or whatever you want to call it, they probably came up with this. You might also see it depicted like this. Here's my anomeric carbon going up. And then here's my ring structure. Now, I bring this up because, well, this is the rare instance where a bond doesn't look like how you typically draw it. This bond kind of looks like a spaghetti bond of sorts, or a noodle bond of sorts. This is a carbon-oxygen bond. Don't get mixed up whenever you see this right here or this right here. Does that all make sense? We're not adding a carbon spontaneously in there. This type of bond is known as a glycosidic linkage. Now, this glycosidic linkage that exists between these two sugars is a 1,4 glycosidic linkage. Specifically, though, a beta 1,4 glycosidic linkage. Now, when you think about cellulose, I want you to think about, okay, well, it's a linear molecule with nothing but beta 1,4 glycosidic linkages. So cellulose does not have any beta linkages. Now, what this affords cellulose the opportunity or the ability to do is, I thought I had a, it allows for a cellulose, like a strand of cellulose, to make a straight line. And then another strand of cellulose, make another straight line. And so it's almost like these fibers or these polymers of cellulose, they just stack on top of one another. Now, whenever those groups stack on top of one another, you've got to account for some other things that are here. What do we know about a hydroxide group and types of interactions, intermolecular forces that hydroxide groups love to participate in? What type of bonding do they like to do? They like to do hydrogen bonding. Now, in an aqueous environment, they can form hydrogen bonds. They can form a network of hydrogen bonds. But also, if you've got one strand of cellulose and another strand of cellulose, can those two strands hydrogen bond with one another? Yes, they absolutely can. Now, has anyone ever seen the Pixar movie A Bug's Life? Okay. I really love that movie. Feel like I'm in a small group whenever I mention this, that's my favorite Pixar movie, but in part because it really had a influential role in my philosophical views of the world. Okay, now with that in mind, there's a scene in it where, okay, who are the villains in A Bug's Life? The grasshoppers. Hopper is one, and then Hopper's got a little brother who's kind of a doofus. Okay, there's a scene where they're hanging out before they go back to steal the ants' harvest, and Hopper demonstrates to his little brother, like, hey, you know, or his little brother's like, let's not steal from the ants, let's not do that. Let's just forget about it. Hopper says, well, you know, here's the thing. We have to maintain an order. We have to let the ants know that they're subservient to us, that they do what we say. So we're going to go and steal their harvest, and they always have to supply that to us. And his brother's like, but why? He says, well, listen, you know, one ant with a wild idea that they don't want to do that, it's not a problem. Kind of like a hydrogen bond. A single hydrogen bond, how strong is that? Is it stronger or weaker than a covalent bond? Weaker. You know, if you think about the magnitude of strength of a covalent bond versus a hydrogen bond, you're talking at least 10 times stronger as a covalent bond. Hydrogen bond 1 is weak, okay? But then Hopper goes on to say, listen, doofus brother, if all of the ants organized and realized their power, what's going to happen? And his brother's kind of like, I don't know. And then Hopper releases this flood of individual, like peanuts or something like that. They're all feeding off of these things. They're all food. And those just smother to near bystanding grasshoppers. So individually, those ants are weak. but lots of them are very powerful. Similarly, cellulose. Well, one hydrogen bond here, not particularly strong. But if you get this complex network of hydrogen bonds, that cellulose molecule becomes very, very strong and it develops a ton of new properties. Because when you have this complex network of them, oh man, that's really hard to navigate through. Which we see that, and we're familiar with that with some of the intensive properties of water. What do you know about the surface tension of water? Well, the surface tension of water from a certain height, it's the same as hitting concrete. If you do a belly flop on concrete, that's not going to feel particularly well. A belly flop onto water from low height, that's fine. High height, it's going to hurt and you're probably going to throw up, if not worse. So cellulose, yeah, it's just a bunch of glucose molecules. But because of that beta configuration, now we have this network of hydrogen bonds. That those hydrogen bonds, can they be disrupted? Yeah, under the right circumstances with, you know, a solvent with a very acidic pH or a very high pH or some extreme conditions like that. But it's kind of one weak force, but lots of them. Those numbers make it so that we can't go outside and take some grass clippings and feed off of those and be like, I got my glucose fill for the day because of that beta configuration of those polymers. Does that make sense? Okay, great. So amylose is another polysaccharide. What I want you to notice about amylose is that down, down, up, down, it's glucose. Repeating glucose monomers has a range, unlike cellulose. This is approximately as few as 300 glucose subunits to as many as 600 subunits. Now, this is one of our major components of starch. So if you go and you eat a potato and starchy vegetables, potatoes or tubers, or something like broccoli or something like that, you're getting a lot of starch. And that starch, well, it's got amylopectin, which has a specified length. And the other major component of it is amylopectin. So amylose and amylopectin are the two molecules that make up starch. Now, with that in mind, if we look at this amylopectin, our other major component, down, up, down, we've got repeating glucose units. These amylopectin molecules are a little bit smaller, 30 to 50 subunits. But the fundamental difference between them is we've got 1,4 linkages, but now we've also bring in 1,6 linkages. So a amylopectin molecule can have 1,6 linkages. There we go. Okay, so when you think about polysaccharides, most of the polysaccharides that you need to be familiar with for this class, as well as to go into metabolism, is think about those those glucose-centric polysaccharides. Cellulose is nothing but glucose, but it's got the beta configuration. Okay, that's good to know. The enzymes that we use to break up glucose polymers are not going to be able to process that. In addition to that, though, we've got that hydrogen bonding network that causes the further problem. Amylose. Can your body digest this? Can cells process this? Absolutely. It's got nice alpha 1,4 linkages. Can you get energy from eating broccoli? You absolutely can. Is there part of it that's not going to be digested? Yeah, because you're still going to have some level of hydrogen bonding that stabilizes this combined with this molecule right here. So when you think about a starch molecule, you've got, or when you think about ingesting starch, you've got something that looks like this. Where my branched molecule, which maybe, you know what, I'm going to do a different color. Here's my branched molecule that constitutes my amylopectin. And now my blue molecule is going to represent changing of the slide. Oh man, there we are. is gonna show up on my rate my professor. Doesn't know how to use PowerPoints. So we've got this network of our blue polymer, our linear blue polymer, nothing but alpha 1,4 linkages. Then we've got our branched alpha 1,4, alpha 1,6 molecule, amylose and amylopectin, which make up starch. Now, can you have multiple of those amylopectins associated with a single amylose? Sure. Think about the length of an amylopectin molecule and the length of an amylose molecule, 300 to 600 versus 30 to 50. So let's say hypothetically, we've got a molecule that looks like that. What's that blue line represent? Amylose or amylopectin? Amylose. Now we've also got maybe something that looks like this. And then something that looks like this. Does that make sense? Okay, so you might have some complicated network like that. Now, with that in mind, there's one last polysaccharide that I want you all to be familiar with, and that is glycogen. These are all kind of nutritional and or structural polysaccharides, but there's one last one, glycogen right here. Now, glycogen is particularly interesting because glycogen is a store or a storage molecule in our bodies that gives us the opportunity to hold on to a bunch of glucose. So your blood sugar is going to be about 5 millimolar at any given point in time. And that's because if you decide, you know what, this week I'm going to go on a diet where I eat nothing but red meat because I saw this guy on Instagram, he's jacked, he's brilliant. And I say those two things because, man, he's super buff. He must be smart. Those two things hand in hand, obviously. I'm being facetious with them. Did it? Of course.
I need that also.
It's the carnivore diet.
Yeah.
And it's like, oh, all that you need is a steak for breakfast. It's kind of like they took Weight Watchers, which was a shake for breakfast, a shake for lunch, and a sensible dinner. And he was just like, how about we change shake to steak? It was like, I'm a billionaire, a brilliant person. So it was, I'm going to eat nothing but red meat, let's say. Well, is your blood sugar going to crash out because for a week you decide to eat nothing but red meat? No, it's not, because your body still has stores of glucose in the form of glycogen. Now, glycogen is a super cool molecule. And for the purposes of this, I'm going to go ahead and open a web browser and go with a glycogen structure. And I'm about to share this for everyone on the Zoom. Here we go. So this right here on the right-hand side of the screen is a glycogen molecule. Now, what I want you to notice is that in the center of it, there's a molecule known as glycogen. Glycogen is a protein that basically glycogen is built off of. But what I want you to notice about this, and I'm going to zoom in. Did not expect that return. Okay, here we go. How about I just click links instead? Is that this molecule, like you can follow the, I'm going to go ahead and follow one of the strings more or less. And let's say that this right here, where I'm just circling, that would be considered my reducing end, where I had my free anomeric carbon that got hooked up to my glycogen and protein. Now, this straight line that I'm drawing, which now I'm going to switch to, yellow. This straight line right here, that linear chain of glycogen is nothing but alpha 1,4 linkages. There are, however, branch points. What type of linkage do you think those branch points are coming from, based on what we saw with amylopectin? 1,6, alpha 1,6 linkages. Now, to show the complexity of a molecule like glycogen, we've got these 1,4 linkages, we've got these 1,6 linkages, and then off of like this straight chain that I'm highlighting and now putting a star by, that's nothing but 1,4 linkages. There was one 1,6 linkage, but everything after that was 1,4 linkages. Does everyone follow that? Here's a branch off of a branch. that branch off of a branch, well, here where I'm circling now in red, that's the source of another 1,6 linkage. So when you think about the abundance of different linkages between, say, amylopectin or glycogen, most of your bonds are alpha 1,4 linkages. There are also 1,6 linkages, alpha 1,6 linkages. There aren't very many of them because what you're going to do is you're going to have a branch and then a straight line after that. Does that make sense? So that's one of the important features of a molecule like amylopectin and glycogen. Now, with that in mind, this molecule right here that you're looking at, well, one, that's a single molecule. It's a polymer, and it's tons and tons and tons of glucose molecules. What does this get you in a marathon? Has anyone run a marathon before or any sort of long race? Around mile 20, what happens? You hit the proverbial wall because you've depleted your glycogen stores, which is why everyone's like, oh, you got to go eat the pasta before the night before so that you got enough sugar. It's kind of a myth, but you know, is there benefit to that? Yeah, because you're going to hang around a bunch of people who are all doing the exact same thing that you're doing the next day. So if nothing else, there's like a psychological component of like, you know, community, brotherhood and everything like that. How do you prevent that? I don't know that there's necessarily a way to like. I think truth be told, I think that it's, I don't know. We'll stop there. And I'm not saying that from, I'm saying that like from, I've run races before and my interpretation is that the more that you train and the more prepared you are, the better off you are on that day. And I think that, you know, there's lots of different things. There are some people who do what's called the maffetone method, where you're running at a low heart rate. And so then you're not like, if you go out for a sprint, you're going to deplete your ATP stores, and then you're going to get into any immediate like supplements of that ATP. If you go out for a sprint and then you're like, I'm going to carry this sprint into a 5-mile run, I'm going to slow down. I've depleted all my ATP. So now I'm going to start processing glycogen or glucose. Once you've depleted like free glucose in your bloodstream, your body's going to be responding by being like, oh, we better break up some glycogen to supplement and to compensate for that. So I think that there's such a mental component to it where you're like, I don't think that I want to start this marathon with a fun sprint. Instead, you're going to be like, okay, I've got 26.2 miles. I think what I'm going to do is I'm going to walk the first 0.2. So then I can say, all right, well, I'm awake. I have 26 miles left. And now I'm going to, you know, steadily increase my speed, or, you know, some people will be like, I'm going to run every single mile is going to be 10 minutes because that's what I can sustain. So that's my interpretation. And then on longer races, you have to supplement by like ingesting something during the race.
Like gels.
Yeah, the gels. And, you know, then you just have to be like, I don't want to have something with so much caffeine that it causes me to have to stop at a Porta-John, because that will be bad. Okay, so what I want y'all to be familiar with, most familiar with, is structures of cellulose, glycogen, amylose, amylopectin. Now you saw that I got to that structure of glycogen. I Googled glycogen structure, and it's on the Wikipedia page for glycogen, so it's a good one there. So all of those, I want you to be familiar with the abundance of different linkages, of different bonds, and what are those different bonds. Those are important things to know. Okay, now, there we go. Now, functional groups on sugars can also be oxidized. We talked about reduction of them yesterday, but oxidation is also something that can happen. Sugars that become oxidized at the aldehyde functional group are known as reducing sugars. Oxidation of the aldehyde group of glucose is the basis of the glucose blood meter test. Glucose oxidase or glucose dehydrogenase convert glucopyranose to gluconolactone. The ring opens, so the carboxylic acid form of glucose can be formed. So gluconolactone. Now, hydroxyl groups of sugars can react with phosphates to produce phosphoesters. We've already kind of seen that with glucose 6-phosphate. Some good examples are glucose 6-phosphate, which is the product of our enzyme hexokinase. Glyceraldehyde 3-phosphate, which is one of our products from the cleavage of that 6-carbon fructose 1,6-bisphosphate. And then we've also got deoxyribose 5-phosphate or D-ribose 5-phosphate. It all depends on what you see at this position. Has that sugar been reduced or has it not been reduced? Okay, hydroxyl groups of sugars. I want you to think about those hydroxyl groups of sugars as being a place where something can happen to them. And that something is either they can be phosphorylated, so that hydroxide group can be replaced by a phosphate. Alternatively, as in the case of something like glycogen, well, that hydroxyl group can be replaced by a polymer of glucose. Now, those hydroxyl groups of sugars can be replaced with amino groups as well. So some examples, or a pair of those examples, would be down here. Glucosamine and galactosamine. Well, glucosamine, we've got down, up, down, but that 2 position, we've replaced that hydroxide group with NH2. Now, galactosamine, is that a sugar that is a wild way to, or a wild thing to kind of predict the structure of? Not really, because, and this is specifically the amine is found at position 2 in this ring. But if we think back to the differences between glucose and galactose, what's the difference between those 2 sugars? They're C4 epimers of one another. Now, glucosamine and galactosamine, those are also going to be C4 epimers of one another. Now, in this particular example, well, this is showing kind of the most common form of glucosamine and galactosamine, where that amine, that NH2, has been added to carbon 2 on that ring. Now, does that mean that you could put together glucosamine or galactosamine where that NH2 is found on another carbon? Absolutely. Would it still be glucosamine or galactosamine? It would. The only difference is, where is that group found? Does that make sense? Okay. Now, with that in mind, we can also have some forms of sugars that are modified in a pretty dramatic form. So here what we have is a sugar that has been modified in a couple of different ways. One, we have a carboxylic acid added to that group. We also have this N-acetylneuramic acid, the linear form of this molecule, is going to cyclize to form this pyranose form of this molecule. Now, this is a molecule that acts as a receptor for influenza viruses. This is for the hemagglutinin protein on the virus is going to bind to this sialic acid. So what I want you to know about this, and between the 2 components of this slide, I would say that this right here is maybe a higher priority. This being the bottom portion of this would be a higher priority for y'all to investigate, just because, and be familiar with, because this is bringing in what I want you to be familiar with. Those hydroxide groups are sites of modification. If you add an amine, well, what's your terminology? If I had mannose as my 6-carbon sugar and I added an amine to it, does anyone want to guess what they would call it? Given that we have glucose that's been aminated, we call that glucosamine. What do you think mannose plus an amine would be? Manosamine. Not too crazy of an idea of a name there. Okay, some common sugars that you should be familiar with: galactosamine, N-acetylglucosamine, where we're taking this same sort of structure, glucose in the middle here, we're aminating it, and then we're acetylating by adding an acetyl group. So N-acetylglucosamine, glucose, if you see that, you should think down, up, down. Okay, we fit the billing there. Amination, which sounds like you're trying to say animation as a 3-year-old. I say that because I have a 3-year-old and that's how he would say it. You're aminating it, you're adding an amine in place of one of those hydroxyl groups, and then you're adding an acetyl group there. Mannose 6-phosphate, well, you're adding a phosphate group to carbon number 6. Now, mannose is not a sugar that I want you to know, or that you're expected to know. What are the 5 aldoses that you're expected to know? Glyceraldehyde, glucose, galactose, and not cellulose, because that's polysaccharide. They're the ones that are highlighted. So now I got to look back, and I was hoping everyone would be like, well, it's these 5, but then I have to look back at my own notes. And this is where someone's like, he doesn't remember them, I don't have to remember them. Touche is what I say to that. Okay, here we go. Here are the 5 aldoses that I want you to know. Glyceraldehyde, ribose, galactose. Oh, mannose, I apologize. Mannose is one and galactose is the other one. Okay, so those 5, the ones in squares. And then ketoses, there's a handful of ketoses, 3 ketoses that I want you to know as well. Okay, so mannose 6-phosphate, fructose 1,6-bisphosphate, which we've already talked about that because that's our doubly phosphorylated sugar that goes through glycolysis. And then glucouronic acid. This is a carbon number 6 that has been oxidized. So rather than having carbon 6 with CH2OH, instead we have that acetic acid or that acetate group. Glycosides, this is our specific term for whenever you have that anomeric carbon and you add O something. That OCH3 is what we indicate right here. And one of the products of a reaction that forms a glycosidic bond is water in addition to the glycoside. So this is our specific term, which isn't too much of a stretch because what were our linkages in our polysaccharides and our disaccharides? They were alpha 1,4 glycosidic linkages or glycosidic bonds. And just so y'all know, if I use the term linkage or bond, I want you to think about those as interchangeable terms. Okay, so when you see something like glycolysis, I want you to think about, well, where have we seen some of these sugars? Glucose, cyclic glucose. If I gave you this molecule, this structure, how would you classify that a little bit more, with more detail? Well, let's look at our anomeric carbon. Actually, let's take it a step. Let's go back a little bit. This is a 6-carbon sugar, so it's an aldose. It had an aldehyde, so it's an aldohexose. When we cyclized it, we formed an anomeric carbon. Now, do we see anything about that anomeric carbon that's significant? We do. The hydroxide group on our anomeric carbon is pointing downward. So this is an alpha D glucose that we're looking at right here. Then if we jump down to these molecules right here, are these 2 molecules both sugars? They are. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. One is an aldose, one is a ketose. If we continue looking through some of these other sugars or these other molecules, we have carboxylic acids as a functional group that you need to recognize. What's happening to these molecules as things are proceeding? Well, how many carbons are we at? We got 3 carbons here, 2 molecules of 3 carbons. And then all the way through here, we still have only 3 molecules or 3 carbons that make up that final product of pyruvate. Now, when you look at this, the things that I want you to know, I want you to know how we would classify glucose versus fructose. Think about, we're taking an aldose and we're making a ketose so that we can make so that we can make 1 3-carbon aldose, 1 3-carbon ketose. Now, what's an important step right here? So glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate is our aldose. Dihydroxyacetone phosphate is our ketose. Okay, so one of each of those is produced. Now, what I want you to do is look at the line for GAP. That GAP goes down to a molecule known as 1,3-bisphosphoglycerate or 1,3-BPG. So then let's look at the line under DHAP, our ketose. DHAP, if we follow the line for DHAP, what happens to DHAP? It gets converted into what? GA3P. It gets converted into GAP, our glyceraldehyde 3-phosphate. So what that means is that does a ketose, or do ketoses move through glycolysis easily, or is the best way to process a ketose by converting it back to an aldose? Does that make sense? So that's one of the things that I want you to recognize about this and think about it as, okay, are ketoses amenable to glycolysis? Yeah, but they need to be kind of reconfigured to make those aldoses. So if you're thinking like, oh, is there a certain kind of priority to aldoses or ketoses? Yeah, that priority is a little bit more towards aldoses than ketoses in glycolysis. So with that in mind, when you look at this, I don't want you to think, oh man, this guy's a jerk. He's asking us to memorize all of this. I'm not. What I'm wanting you to do is to be able to apply what we've looked at to this. Does that make sense? Are there lots of nuances and subtleties to this pathway? 100%. And that's reserved for metabolism, where you'll dig deep into that. What I'm hoping is that from this class, when you go on to take metabolism, you'll be like, aha, I've got a little bit of a leg up because I understand this. Cool? Great. Okay. So now we're going to do our kind of transition into nucleotides and nucleic acids. So I'm going to look at my phone for a second. Okay, there's my reset. All right, so we're back. We're talking about nucleotides and nucleic acids. The first part, and the reason that I like to talk about sugars, then go into nucleotides and nucleic acids, is because we have a little bit of a foundation for this. And that foundation is in the form of, well, when we talked about ribose being converted to deoxyribose. In ribonucleotides, the pentose sugar is ribose. In deoxyribonucleotides, the pentose sugar is deoxyribose. So when you hear or think about nucleic acids and nucleotides, I want you to think, okay, we need a pentose. We don't need a hexose, we need a pentose. Now, ribose. We look back at this. Ribose as a pentose, it is an aldopentose. Ribose is right there, not to be mistaken for ribulose. Okay, so ribose is our aldopentose, which I don't think that I'm sharing with everyone. Yeah, no, I am good. Our ribose is an aldopentose. Now I'm going to go back to this right here. So what do you think that means about deoxyribose? Is that also an aldopentose? It is. So that's our 5-carbon sugar. Ribonucleotides are going to have this on the left-hand side, and deoxyribonucleotides are going to have that right there. Now, one of the things that I want to familiarize y'all with is the numbering scheme. A ribonucleotide has a base, a phosphate group, and a 5-carbon sugar. That 5-carbon sugar is ribose. Now, our numbering, when we talk about nucleotides and nucleic acids, goes from carbon 1, 2, 3, 4, and 5 to 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. That prime is to distinguish it from the numbering on something like the base that is going to, the nitrogenous base that is linked up to that carbon 1 prime. So if you've taken a biology class, whether it's a cell biology or an intro biology class, you've probably talked about DNA replication or DNA synthesis. You've probably talked about 5' to 3' directionality. And it's entirely possible, if you're anything like me, you heard that and you thought, aha, I've memorized that, don't need to know any specific information about that. And then you get to this class and you say, oh, that's where the 3, oh, okay, that prime, it wasn't just like, here's a fun word to remember. So when we talk about a deoxyribonucleotide or a ribonucleotide, we're focused in on and centered on the 2' OH group. Does that make sense? Okay, great. Now, here's some terminology that's important to kind of distinguish. A nucleotide versus a nucleoside. A nucleotide is composed of a nitrogenous base, a sugar, and at least one phosphate group. A nucleoside, by contrast, well, it doesn't have those 3 components. A nucleoside is going to have a sugar and sorry, I can't control Z that. Oh, right. A nucleoside consists of the sugar and the base only. So a nucleoside lacks a phosphate group. I personally always remembered that because that T, phosphate, there's that hard T in the word phosphate. Nucleotide needs a phosphate. Maybe that's unnecessary, but that's how I remembered it. So nucleoside is a sugar and a base. A nucleotide is sugar, base, and phosphate group. Now, the key or one key there is at least one phosphate group. Commonly, if you think about adenosine triphosphate, how many phosphate groups does that have? 3. Where are those 3 phosphate groups more than likely found? Which carbon? Probably on the 5'. Could they be found on the 3'? Yeah. That would be a 3' nucleotide. Are those very common? No, they're not. So there's some conventions that we kind of align with like, okay, this, you're basically never going to see this molecule, whereas this is very, very common molecule. So we're just going to use that as, you know, ATP. The implication when you hear ATP is that it's 3 phosphates on the 5' carbon of our sugar. Now, nucleotides, when we think about a nucleotide, those phosphate groups, since they're typically found on our 5' carbon, we make a chain of them. They are our alpha, beta, and our gamma phosphate groups. That 3rd phosphate, the one farthest from our 5' carbon, is our gamma phosphate. Now, it's pretty common to have a NMP, NDP, or NTP. That N can be replaced by, well, the name for that nitrogenous base. So if you saw ATP, one thing that's important to recognize about ATP is that the implication there is you are dealing with a ribose or a ribonucleotide compared to lowercase d ATP, where you are dealing with a deoxyribonucleotide. So anytime you talk about, oh, I'm going to make DNA, do you need ATP or DATP? If you're making DNA, you need DATP. You need a molecule that has been, that the hydroxide group from the 2' position has been removed. Does that make sense? So when you think about processes that, like what we were talking about with glycolysis, generating ATP, what's the sugar in that ATP that's generated from glycolysis? Is it ribose or is it deoxyribose? It's ribose. There is significance to that lowercase d or not being present. Does that make sense for everyone? Okay, cool. Now, our nitrogenous bases, they fall into 2 major categories. pyrimidines and purines. Pyrimidines are 6-membered rings, and their numbering scheme looks like this, going in a counterclockwise or in a clockwise fashion, I'm sorry, around that one ring, starting at this position right here, this nitrogen 1. Now notice that doesn't have prime. Why do we have those prime, or why do we not have numbering of 1 prime, 2 prime, 3 prime? Well, because we have that numbering when we talk about our sugars. So if I talk about atom 1 prime in a nucleotide, it's implied that I'm talking about that carbon 1 prime in my ribose or deoxyribose. If I talk about the atom number 5 in a molecule or in a nucleotide, well, then I'm talking about that right there, as opposed to my 5 prime within my sugar. Does that make sense? Okay. So my pyrimidines are single rings, and those single rings are cytosine, thymine, uracil. My purines are adenine or guanine. Your numbering for each of these is going to follow this convention, where we have our purines. This is 1. This is 1. This is 8. This is 8. Now, the atom that is going to connect a pyrimidine to our ring is going to be nitrogen number 1. That atom that connects our purine to our sugar is number 9. So if we talk about a nucleotide, so we jump back right here. If we talk about something like ATP, A or adenine, that is a purine. So the linkage that connects this atom right here, so let me jump back a second, that nitrogen to that right there, there's a couple of things that you need to know. One, it is a glycosidic linkage. The only difference is this is an N-glycosidic linkage. Now, when we want to classify or give more definition to a glycosidic linkage, we say 1,4-glycosidic linkage, right? Okay, so if we have ATP and we have an adenine up here as our base, that's nitrogen number 9. What is this atom right here that I just underlined? It's a 1 prime. So we have a 1 prime 9 N-glycosidic linkage. Does that make sense? Now, when we jump over here and we talk about the pyrimidine, we have a 1, 2, 1 prime N-glycosidic linkage. So here's that's my 1 prime carbon. This is 2 prime, 3 prime, 4 prime, and then CH2, OPO3, 2 minus. Does everyone follow that? So it's a 1 prime to 1 glycosidic linkage, or it's a 1 prime to 9 glycosidic linkage, N-glycosidic linkage. So 1 prime to 9, that's a 9, and then 1 prime to 1 N-glycosidic linkage. Is everyone good with that? Okay. So these are our bases. What I want you to know about these bases is the differences between them. I'm going to be asking you questions about, simply identifying them. I think that it's helpful to use this scheme right here. I'm going to erase everything on this slide. I said pen. 1, 1, 1, 2, 2, 2, 3, 3, 3, 4, 4, 4. So our three pyrimidines are cytosine, thymine, and uracil. Cytosine at position, or what makes cytosine different from thymine? Well, at position 2, is there any difference between those two nitrogenous bases? There's not. 3, well, the only difference is that we've got a carbon nitrogen double bond for cytosine, and then a single carbon nitrogen bond. Thymine, we have a hydrogen going to that. Do both of those nitrogens in position number 3, they have 3 bonds. What does that mean about nitrogen? Does it have a charge? Is it neutral? How is it neutral? What else does what does nitrogen really love to have? 3 bonds and a lone pair of electrons. So this is a place where I know that folks have a tendency to make a mistake. They like to draw this nitrogen to carbon bond, and they like to draw it as a double bond. And then when they get to drawing cytosine versus thymine, they'll say, okay, well, I've got my nitrogen to carbon double bond, and then I got to throw a hydrogen on there. No, you don't, because when you do that, what does that mean for that nitrogen? It has a positive charge. So don't fall into that kind of trap. Distinguishing between cytosine and thymine, well, at position number 4, cytosine has a double bonded oxygen, whereas, thymine has a double bonded oxygen, cytosine has an amination. At position 5, we have a methyl group on thymine, and in cytosine, at positions 5 and 6, we just have a double bonded carbon. So CH double bonds across there, those are your similarities and differences. Now, are those differences significant? Yes, they are, because they enable cytosine to have 3 hydrogen bonds with guanine and make thymine only have 2 hydrogen bonds with adenine, its bond pair. So I want you to be able to decipher the differences and similarities between these two and know those numbers, know the numbering scheme. So if you can recall that, that's going to be very helpful. I think that it's helpful to start with, well, how do I get connected from my nitrogenous base to my sugar? I think that's a good place to start out. Now, these bases, the keto forms of these bases can predominate. Both sets of base pairs are going to occupy the same geometrical space. And so that allows for these bases to stack on top of one another to give us what we now know to be the double-stranded structure of DNA. Now, our nomenclature. We've talked about nucleoside versus nucleotide. Lowercase d indicates that we have a deoxynucleoside or a deoxynucleotide. So our primed numbers, 2' refers to a specific atom within the ring of my sugar. Our non-primed numbers refer to the base. Now, this is something that is unique when we're talking about nucleosides and nucleotides and DNA. If I'm asking you about the structure of ribose alone, independent of a phosphate or a nitrogenous base, I'm not asking you about it being a 2' carbon. That would be a carbon number 2. Does everyone follow that? So it's only when you add the base do you run into the problem of like, okay, I got to have a prime designation as opposed to no prime. Cool. Okay. The phosphate group may be attached to the sugar at either of 2 positions. If at C3', it's called a 3' nucleotide. If at 5', it's called a 5' nucleotide. We have our deoxyribonucleotides. On the top, we've got our ribonucleotides, and on the bottom, we have our deoxyribonucleotides. Now, one thing that you'll notice that I do, and I think many other people do, is I would say more times than not, people are going to not use this nucleotide name to to basically describe the nucleotide corresponding to adenosine. Does everyone follow me there? So when it comes to names and it comes to naming, if I'm talking about a molecule of ATP, if I'm talking about a molecule of ATP implying that my phosphate group is on the 5' carbon, I'm actually not going to say adenylate. And I'm not going, what I'm actually going to say more times than not is adenosine 5' triphosphate. So adenylate indicates that you have an adenosine and you have at least one phosphate. Does everyone follow that? So more times than not, you're going to use the nucleoside name, adenosine, guanosine, uridine, cytidine, deoxyadenosine, deoxyguanosine, deoxythymidine, deoxycytidine. And then to that, you're going to add 5' triphosphate, 3' triphosphate, 2', yeah, less common, but 3' diphosphate. Does that make sense? So what I would recommend is nucleoside plus how many phosphates there are. Now, with that in mind, for this right here, what you need to be most familiar with for names and abbreviations, single letter codes A, G, C, T, and U correspond to, well, what is our nitrogenous base? This is something that, if you're fortunate, you've seen Jurassic Park, and so that may be where you got this from. But the lowercase d that you put in front of any one of those letters is going to say we're talking about a deoxy version of this. And that means that we lack the hydroxide group on the 2' carbon of our sugar. Now, monophosphates or monophosphorylated nucleotides can exist in a couple of different ways. We could have adenosine 5' monophosphate. Notice this is the nucleoside name plus where is that phosphate group, and mono to indicate one phosphate. Adenosine 2' monophosphate. The structures are basically the same, but this is a 2' phosphorylated molecule. This is a 5' phosphorylated molecule. Adenosine 3' monophosphate. And then if you've taken any sort of physiology class or you've talked about a molecule known as CAMP, cyclic adenosine monophosphate, or CAMP, that's a secondary messenger that's involved in tons of different processes. If you drink coffee, well, and let's face facts. Anytime someone mentions coffee, no one is like, oh, decaf, of course. Instead, if you drink coffee, you've accepted that you're drinking something that does not taste good, and you really just need it for caffeine. It's kind of like beer. At any rate, when you drink coffee, that caffeine, well, that is a molecule that is not going to be able to cross any sort of membrane. So what that's going to do is that's going to be received, and then that receptor is going to stimulate the production of this cyclic adenosine monophosphate, which is going to turn on activation of different genes, regulate different proteins. And this right here is an example of cyclic AMP. Now, cyclic AMP could also be a 3', 5' cyclic monophosphate, in which case that phosphate group is kind of buckled underneath that ring structure. It's possible. But this designation right here shows we've got a phosphate group that we have 2' and our 3' carbons where a ring results from that. Does everyone follow that? Okay, cool. Now, with that in mind, nucleotides, nucleosides, they are important for, just like the production of ATP, but you're actually also going to see them in a number of other settings. Coenzyme A, and then this molecule down here, nicotinamide adenine dinucleotide, and then another molecule, flavin adenine dinucleotide. Let me give you a little bit more context for all of these. If you're familiar with the Krebs cycle or the tricarboxylic acid cycle, this molecule right here, coenzyme A, is responsible for helping you get those carbons from glucose that we just broke down into pyruvate, this coenzyme A is helpful for getting 2 of those 3 carbons from pyruvate into the mitochondria or into this citric acid cycle so that we can fully oxidize that. Nicotinamide adenine dinucleotide. This exists in 2 different forms. NAD plus and NADH. So we have an oxidized and a reduced form of that. And just so y'all know, whenever we talk about oxidized and reduced forms of something, I want you to go back to, like, this is a redox cofactor existing as its oxidized or its reduced form. If you go back to Gen Chem 1, whenever I teach Gen Chem 1, I use very basic examples like this, and I'll say A goes to A plus. In order for A to go to A plus, what had to happen to A? It had to lose an electron. So you could say that A plus is my oxidized or reduced form of A. Well, it's A, but it lost an electron, so it's the oxidized form. You could also say that A is the reduced form of my molecule. Does that make sense? So when you see something that this right here applies to this. So NAD plus, is that my oxidized or is that my reduced form of this molecule? That's my oxidized form. NADH is my reduced form. I just bring this up because I feel like my learning journey, whenever I got to organic chemistry and we started talking about oxidation reduction, it was a little bit more complicated. And then my ability to pick out the oxidized and reduced form, I struggled with this quite a bit. And so what I want you to do is think about, if it's got a positive charge, that's my oxidized form of that molecule. If it does not have a positive charge, that's my reduced form of that molecule. Keep it simple for yourself. Okay. Yeah, we're nearly done. And good. Today and tomorrow's lectures are probably going to be a little bit shorter, but don't worry, we're going to sit in silence as soon as I'm done in lecture, and we're just going to sit. We're going to ride out the awkwardness of a silence. So when we talk about a nucleotide, we have an N-glycosidic linkage that connects our base to our sugar. Another very important linkage or bond that exists in a DNA or an RNA molecule is known as a phosphodiester linkage. So this phosphodiester linkage, well, let's see. We've got a phosphate, so phospho, done. Diester, oh, rats. Well, let's see. There's one ester, and then lo and behold, there's another. So phosphodiester. At the heart of that, we've got a phosphate group, and then we have 2 esters coming off of this. Now, another type of bond that I want to bring up is that you will probably hear is this bond right here, existing between our 2 phosphates, or between phosphate groups. So this right here, that bond is known as a phosphoanhydride bond. This is also a phosphoanhydride bond. This right here is a phosphoester. When you think about DNA stability, I want you to think about phosphodiester linkages, phosphodiester bonds. Are there other types of bonds? Phosphoester, yes. And phosphoanhydride are other bonds. And I bring that up just because I want you to think about, like, triaging information. What am I most likely going to ask about? If I give those 3 options, I'm probably going to ask, can you identify and do you know what a phosphodiester linkage is? Are those other 2 possibly going to surface? Yeah, possibly. But if I don't ask about a phosphodiester bond, I wouldn't expect to be asked about a phosphoanhydride or phosphoester bond. Does that make sense? Okay. So when we talk about the primary structure of a molecule of DNA or RNA, well, these are molecules that are linked 3' to 5' by phosphodiester bridges. Oh man, I'm just a jerk because I said linkage and bond. I'll use those 2 interchangeably. And now I bring in bridges. Oh, that might be the last time you see bridges, but nevertheless, that's an interchangeable term. I think everyone kind of got that from context clues. Ribonucleic acid and deoxyribonucleic acid. What are we differentiating between those two? The 2' OH group. The sequence of a DNA and RNA molecule is always going to be read in a 5' to 3' direction. In terms of genetic information, this is going to translate to a protein, and this is going to correspond in N to C directionality of a protein. So we'll talk about primary structure of proteins, secondary, tertiary, and quaternary structure as well. But we're reading a DNA molecule in the 5' to 3' direction. And so anytime you see a DNA sequence of A, T, G, C, the implication there is that this is in, well, the 5' end is A, 3' end is C. Deal? Okay. Now, here what we have, I got to make sure that I'm not concealing anything up there. Good. We have a molecule of, let's see, is this DNA or RNA? I've got to vote for RNA. Based on what? And the OH at the 2' position. Samantha, I think you joined in, you said that on Zoom and I caught that. But yes, the 2' OH group, by virtue of that 2' OH group being there, that's our first clue. That should be our first tell that we're talking about an RNA molecule. This molecule reads in 5' to 3' direction, A, U, C, G. U is uniquely reserved for RNA. And when we look at this molecule, there's one other detail that we need to pick up on. This right here, this molecule is phosphorylated, has a phosphate group at that 5' position. Does a molecule have to have a phosphate group on the 5' or 3' end of it? And if so, would that be a way of saying like, oh, it's not DNA or it's not RNA? No, because at this end, if we don't have that, and we don't have that, is it still a molecule of RNA? Yes, it is. The key is though that it's not a phosphorylated molecule of RNA. Now, with that said, because we do have those, we're going to kind of call that out. And we're going to look at it. And we've got two different ways of looking at it. One of them, to me, is kind of silly, and it's kind of overkill. Because one of the things that we know, in order for these bases to be read in a sequence, what do we have to have for those bases to, be a continuous molecule? We have to have that phosphodiester linkage between the sugars. Does everyone follow me there? So we got to have these, and these. These phosphates correspond to that. Does everyone see that? I think that's a little bit silly because that's almost like saying, well, I personally live in San Marcos, Texas on Earth. I gathered that it was Earth when you said Texas, among other things, but it's kind of overkill to me because when you see AUCG, there's not really any other convention or any other way that you would see AUCG as a part of a polymer that does not include those sugars and does not include those phosphodiester linkages. Now, with that in mind, there is one part of this that is important. That. The 5' phosphate, indicating that is important. You could have a nucleotide that looks like this, A, T, C, G, G, P. The one that I just wrote there, what does that indicate in terms of, I'll ask a couple of questions. Does this molecule that I just wrote, does it have any phosphodiester linkages? Absolutely. Does it have a 5' phosphate? Well, and I don't want that to be like a confusing question. Yeah, there's a 5' 3' phosphate in the form of a phosphodiester linkage, but I'm talking about the termini. Is there a 5' phosphate on the termini of this molecule or on the terminus? No, it'd be right there if it was. Is there a 3' phosphate group? Yes, there is. So whenever I write something like this, ATC, that's a molecule that's phosphorylated on the 5' end and the 3' end. Now that's important because when you have a molecule that's phosphorylated on one end or another, then it's got an ability to react with some molecules, but not with others. So if I were to show you this and then say, I'm going to add a phosphate group, right? I want to add, just in terms of the enzymes in our cells that do this, I want to add this A to the end of this. That's actually going to be a problem because what we need in order for this to happen is for there to not be that phosphate group there. Now, there's a class of enzymes that does that, and we'll get into that later on down the line. The enzyme that's responsible for linking up pieces of DNA is known as a DNA ligase, but that's not for right now. Now, the next thing is, if this is a deoxyribonucleotide, well, then we got to throw that lowercase d in front of our molecule. So if I write PATC, that is an RNA molecule. That is a deoxyribonucleotide. Now, can you get something that's kind of clunky and funky like this? Yeah. That would be a molecule that consists of a deoxynucleotide as well as 2 ribonucleotides. Because I've designated, well, the A is a deoxy and the T and the C are ribose sugars. Does that make sense? Okay. DNA double helix, which thankfully, due to the work of Rosalind Franklin, people see this structure. The major components of this structure, it has a major groove and a minor groove. The way that this molecule kind of contorts itself gives these kind of large regions. Now, the cool thing about this is that this enables enzymes to basically scan along, basically bind to this molecule and scan along it and see within these major and minor grooves and detect, oh, well, that's a G and a C, that's a C and a G, and basically read it in some form or fashion. The exterior of this, and if anyone's ever heard this, the overall charge of a DNA molecule, that overall charge is what? It's a negative charge. Why is that? Because of this green ribbon, that is kind of our boundary, our outside of it. This is our sugar phosphate backbone. Each one of those phosphate groups bringing a negative charge, giving us a molecule with a kind of, it's almost like it's got a coat of negative charges surrounding it. Now, with that in mind, when we think about those nitrogenous bases, did we see any formal charges? No. They're really good at hydrogen bonding. They're not really good at any sort of ionic bonding. The bases, they're hydrogen bonded inside, and those backbones, these 2 green ribbons are anti-parallel to one another, meaning that one reads in the 5' to 3' direction, the other one reads in the, well, going in the opposite direction, the other. So it would be the 3' to 5'. Our base pairs, I mentioned this, but here I'm showing it. A loves the hydrogen bond with T. G loves the hydrogen bond with C. Adenine and thymine, A to T. That's 2 hydrogen bonds. G to C, that's 3 hydrogen bonds. I think that this is a good model to visualize and try to like wrap your mind around it and think about the atoms that are involved in this bonding. Because when you do a quick glance at this, it's like, oh yeah, I got it, done. But then if you were to look at it and say, all right, well, which atoms are actually, forming that bond? Well, then I think that it's helpful to go back and say, okay, this is carbon number 1, right? Or this is nitrogen number 1. Then where do I go? This is 2, 3, 4, 5, 6. Okay. Then it's good to take this and jump back. Say, all right, well, my numbering, I feel decent about it, but there's still some room for improvement. I feel good about my numbering of my pyrimidines, but what about my purines? Okay, so that nitrogen on my 6-membered ring of my purine is going to be 1, and I'm going counterclockwise. So I'm gonna jump back here in just a second. Okay, so this is going to be 9, because that is my N-glycosidic linkage to my carbon 1 prime. This is going to be 8, 7, and then my nitrogen 1, and I'm going counterclock, or now I'm going clockwise because I'm looking at the opposite of it. It'd be easier to just do that. So I've got. Okay, so this is going to be 2, 3, 4, 5, 6. Does that make sense to kind of like look at those two figures and like apply that information? So this gives you a way to see like, okay, what are the groups that are interacting that cause that hydrogen bonding? Now, with that in mind, G and C, you got 3 hydrogen bonds. A and T, you got 2 hydrogen bonds. As we all know about a bug's life, 2 hydrogen bonds are not as strong as 3 hydrogen bonds. Now, when we talk about a DNA molecule that has tons and tons of G's and C's, but not quite so many A's and T's, can you think of, well, what does that mean for the strength of that molecule? If you've got, let's say, a molecule that consists of 70% G's and C's, and then another DNA molecule that's 50% G's and C's, which one of them is going to be stronger or is going to be held together by more hydrogen bonds? The one that's 50% G's and C's or 70% G's and C's? 70% G's and C's. Makes sense. Now, there are organisms on this planet, maybe on other planets, we don't know, that they have a high GC content. That high GC content of their whole genome translates to stability. One example, if you've taken a biology class, you probably learned about the polymerase chain reaction, PCR. Thermus aquaticus was the organism that enzyme was ripped out of. Thermus aquaticus thrives in a thermal vent where it's extremely hot, there's a very high pressure. Thermus aquaticus has a higher GC content than we do. The number of hydrogen bonds that hold its genome together is greater than the number of GC bonds that hold our genomes together. If you took any of my genomic DNA, or, you know, I'll make this relatable, any other person in this room's genomic DNA and put it in a thermal vent, what's going to happen to that genomic DNA? It's going to be denatured. It's going to basically melt into a bunch of different pieces. Thermus aquaticus, by contrast, it's not going to have quite that same issue. We have something known as a Chargaff's rule, which is based on Linus Pauling's or Watson and Crick's base pairing, A to T, G to C. And yeah, that's our stability. Okay, RNA structures. There's tons of different unique RNA structures that are a little bit different from that of DNA. And the reason for that is RNA is predominantly a single-stranded molecule. RNA is a single-stranded molecule. It forms these little funky structures and it forms these little helices, whereas DNA is a little bit more straight and narrow. It likes to form a double helix, and that's about it. RNA, because it can do the same thing, is also less stable than DNA because it can hydrogen bond with anything. It can hydrogen bond with water. And as a result, whatever it hydrogen bonds with, it can be frayed, it can be destroyed because of that. DNA, if you took DNA and you put it in a microcentrifuge tube and you put it on this desk and you left it there for 50 years, you can come back and probably still see that it's DNA. RNA, by contrast, it's toast in a minute. If you ever get the opportunity to work with RNA, excuse my language, but it is a pain in the ass. I'm sorry for swearing, but that it just takes me back to any times I had to do RNA extractions as a graduate student, because RNA loves to hydrogen bond with everything. And if you do it more, it's the sort of thing where if you do it more often, you get better technically with it. But I hated doing RNA extraction so much, I was like, I'm gonna do one like every 2 years. So anyway, RNA can form all of these different structures. And it's because it's single stranded. Okay, and then I think we're gonna go ahead and stop right there. So, I'll let y'all go 11 minutes early. We'll get it back tomorrow. I'm gonna stop my recording right now. And I will take off with this slide tomorrow, this last slide. And yes, I do.